A lot of people here are very excited about building systems like this. I am one of them, and I love it. And I love to see what is around here at the conferences, at this conference. Let me get this to work. Yes. And I think there's a very interesting role for light microscopy to fulfill in the context of biology, which is that I think it can provide in situ, in cells, stoichiometry and conformations, and as well as in vitro dynamics, and why not as well as conformational dynamics in live cells. But to get to that, we need to do a bunch of things that are challenges. Like we need targeted labeling, we need high localization precision, 3D, as Jörg showed. Even here, it starts to disappear now. Isotropy, and of course, targeting multiple species in a sample. And I think that with a lot of expansion microscopy is doing great, RESI, and of course cryo-electron tomography and other techniques. But I think there's a key role to play for light microscopy in the context of looking at flexible, inhomogeneous, and dynamic objects, because these are hard to catch by frozen techniques like single particle cryo-EM. And in my lab, we're pursuing this type of goals, looking at conformational changes of proteins, as well as flexible objects like nucleic acids. Jörg introduced all the relevant aspects of SMLM, but I want to say a bunch of things, because one of the challenges in the single molecule localization techniques is the precision that you can get, how many photons you need for that. And what I'm going to talk about is MINFLUX localization, which attacks that problem. So single molecule localization, for the purpose of imaging blinking fluorophores or for the purpose of tracking, has a common element, which is the localization precision, that is proportional to the wavelength of light, inversely proportional to the numerical aperture of the lens, and also has the number of photons there. The number of photons there is because the more signal you got, the best fit you can have of your data to a model of the emission PSF. Now, there's an alternative to this that came up and was named MINFLUX, where the localization precision doesn't need to scale directly at first order with the wavelength, but it can scale at first order with a geometrical parameter that you can tune and manipulate. And you can even put an exponent here on the number of photons that is greater than 1. So that's very exciting. You may have heard about this technique, and you may have seen explanations that involve the following. Instead of using a uniform illumination, you illuminate your dye or your sample with a pattern that has some structure, and particularly a 0 of intensity, and you try to put your molecules very close to these zeros of intensity, and somehow this leads to a better precision. This is a nice approximation, and then you build an image molecule by molecule. This is a nice approximation to understanding what's going on, but there's a lot behind it. And I want, one of the take-home messages I have for you today is to know how this works, why is this better. So let's analyze first the problem of localizing a single molecule. Let's say I have that molecule and I illuminate it with a pattern that has a 0 of intensity, and I collect some photons. Then I move my beam and I collect again some photons, and this is a sequential experiment, 2 steps. And from the number of photons that I collect, I should be able to pinpoint where this molecule was. There is an analogy to this, where you can think that you have created a ruler where the size of the ruler is the distance between the 2 zeros of the sequential beams, and the number of photons is the number of divisions. There we go, the number of divisions on this ruler. So what do I mean by this? If you were to collect 4 photons in total and your molecule was here, you would first collect 0 photons and then all the rest. If your molecule was here, you would collect the 50-50 case. So every point in space, or many points in space, get labeled with a photon combination that you can measure. If you happen to collect more photons, speed up, you're gonna get more combinations of photons distributed in the same distance. So each piece of space or each measurement would correspond to a smaller piece of space. And that's a way to think of how the precision arises from using more photons. But of course, we don't want to use more photons, right? This is not what I said. what you can do is keep using few photons, but manipulate this ruler by shrinking it and using these two sequential beams, but closer to each other. So this is pure geometrical, and of course, at the end of it, you can have a few photons, but still your measurements are very closely spaced anyway. So you can have high precisions with few photons if you modify the geometry of your problem. And this is what you're really doing in MINFLUX. So this gives you a bit of a flavor of why this scales with a geometric parameter directly, and this geometric parameter is the distance between these sequential beams that you used. If we put some math on it, the numbers of photons that we collect are modeled as Poissonian emission on a given exposure time, and their mean is proportional to the intensity that is exciting the molecules. You can do this twice, and it's interesting to look at this ratio, this fraction of photons, so how many photons you got in the first versus the total, and this is going to be proportional to the ratios or fractions of intensities. So if you happen to measure 60 and 40 photons, you have a ratio of 0.6. You can come to this equation and find x, the position. If you do this, you're doing maximum likelihood estimation. And this number, of course, is random. You could have measured 37 and 63. So this follows a distribution. In this case, given the way that it's written, is a binomial distribution. And this distribution gets mapped to the real localization axis by this function p that is fully dependent on the beams. So this is how the photons that count in statistics get mapped to the spatial localization or estimation statistics through a function that is fully dependent on the beams. So we can touch the beams, we can touch this function. And this is where the trick is. So we care about how narrow this distribution can be for a given number of photons. And one can calculate the minimum size or the minimum standard deviation you can have by using a statistical tool or quantity called the Kramer-Rao bound. It's a bound of how narrow this can be. You can compute that as the inverse of another quantity called the Fisher information, which is the average curvature of the likelihood function of the problem. That is a mouthful, but what matters here, and anybody who's developing methods methods that you want to improve, you may want to make them more precise or more economical or whatever, I do recommend that you look into these quantities, because if you can write the direct problem of the situation that you're working with, you can write the probability of measuring what you measure as functional parameters, then that's what you put in there, just the direct problem, and only with that and computing this thing, you can know the bounds of your technique, and you can know if what you're pursuing is worth pursuing. So if you plug the situation that you have for this localization strategy, you end up having this very innocent quadratic function as a function of the position. And if you evaluate it at the origin, surprise, surprise, the wavelength is not there because I never put it, because I didn't need it. But comparing it with the camera localization or STED resolution expressions, there's, of course, this parameter there, which means that if I shrink it, I can have more precision for free. How to interpret this? Well, I told you about this ruler. So if you shrink this parameter, it means that this ruler is smaller. And what it means is that the function that is mapping photon statistics to spatial localization gets sharper. And that's how you get the precision. And the Kramer-Rao bound goes in agreement with this shrinkage of distributions. But the trick is there. We call this MINFLUX, not for minimal photons fluxes necessarily, but for me, my interpretation is that the M of MINFLUX is maximally informative luminescence excitation, because we are manipulating the Fisher information through patterning the excitation to maximize, if we can, the Fisher information of the localization position. Very interestingly, anything that depends on space is divided by this L, is adimensionalized. And that is because if you shrink the distance between beams, zoom in and increase your power, the function that is mapping the statistics remains invariant. So there's an invariance to spatial scalings, and that allows you to manipulate the information even further. And we'll talk about that in a moment. But with that, I close the harsh part of this. We're gonna speed up, but not so deep. So bottom line, remember this. If you look at the performance, of course you can make measurements, look at precision versus number of photons. The precisions that you get are in line with the Kramer bound. This is not a fit, this is just an overlap of points over the bound. You can get more precise, and this is overall tunable depending on the problem. You can keep shrinking your pattern, but of course, in the absence of background, everything's great. When you have background, there is an optimal place where you should be, because at some point you're just overlapping constant profiles, so it means nothing. And of course, the precision is position dependent, and it is anisotropic, meaning different positions in space for a molecule will give different ellipticities. What can you do with this? Well, if you have blinking molecules, you can have little bursts of fluorescence. You can multiplex positions of your beams very, very quickly and create these time traces where each trace corresponds to the photon collection that you get from every one of these points. And if you just localize each one via this maximum likelihood approach that I mentioned before, you can just make a 2D histogram. It looks great. So you can use hundreds of photons for 1 nanometer or so precision. Again, this is tunable. If your molecule moves, you can transform your photon traces into trajectories, and you can do tracking. What you see there is an origami that can oscillate, and here you see the X and Y coordinates. And this was tuned for high precision and temporal resolution. And finally, if your molecule gets out of a good region, you can put a feedback loop and keep it there. And that's what we did to track the small and large ribosomal subunit proteins in E. coli with photoconverter proteins. This was actually the first experiment we did with MINFLUX. So live proteins background, not the greatest choice. It would have been faster otherwise. But for the same photon budget that you have, or say for a fixed photon budget on this type of probe with camera localization, you get with MINFLUX 100-fold more points in your trajectories, which means that for a single molecule, you can even get a trace of diffusion coefficient as a function of time. You can do a lot with that. And this was tuned to have very high time resolution, not so high spatial resolution, but it depends on what you want. But this was localizing with only 10 photons. What strategies are there to use this technique? Well, I'll show you this, but of course you can go 3D. If you go 3D, the nice thing is that this ruler story that I told you is also true if you look at the axial direction. So if you have a beam that has a 0 across the axial direction, you can do the same thing, and you can decouple the axial from the lateral precisions by tuning the distances or exposure times of these beams, which is great. And finally, you can, of course, use a pattern, collect some photons, localize, then recenter, use another pattern, and so on, in a few steps. And when you do that, you can catch molecules across an entire confocal volume, because remember, everything's very tiny here. And if you can cover a full confocal volume, meaning a full volume where photoactivation might occur, then you can do cells. The interesting part here is that the precision scales with the number. So the photons have a power that is the number of iterations that you make. There are limits to this. It's not magic, but it's another manifestation that you're making a better use of the photons information-wise. To do images, you can photoactivate. You can do this iterative approach, and then this is in a few milliseconds, and then squeeze out all the remaining photons with the most efficient pattern that you can, and then produce a localization. You can do this in a mosaic way throughout a sample and imaging cells. What you see here are nuclear pores that we saw before. In the storm modality, you can do this with photoactivatable proteins as well. Looks very nice. And you can even use, of course, the nice thing, as Jörg mentioned, is that you can do this live, and live looks pretty good as well. For multicolor, you can have just 2 channels and use spectral ratios to classify one molecule or another. These are the MINFLUX counts, meaning the sum of all your counts, and these are the spectral counts. So you can see that MINFLUX count give you a shape, and then you can produce a histogram of spectral ratios. They look quite good because the signal to background ratio is good, and then you can totally separate what is what and color your sample, and this happens quite well for cells as well. So you can do multicolor. There are other ways to do multicolor. This was a storm version. And for 3D, of course, we looked at the nuclear pores in 3D. Actual precisions were higher even than the lateral ones because we didn't account for some details, so we even boosted more. And here you have another example of PSD95. Let's see if it gets there. Yeah, PSD95 scaffolding protein of the postsynaptic side looks very nice, and you can do a lot with that if you're into it. Now, I'm talking about precision. So what am I talking about? This is important because in this field, you hear a lot about it. I'm talking about precisions which are bounds for standard deviations of distributions. I might say, okay, these distributions are Gaussian, but keep in mind that for a one-dimensional Gaussian distribution, a width of sigma represents 38% of the distribution, while for a 3D Gaussian, a sphere of diameter sigma represents 4% of the distribution. So in the field, we're always talking about this number. It's fine to talk about this number as long as we all know what we're talking about. So keep in mind that then if I say that in 3D, I have a 1 nanometer precision, I'm talking about the sigma. And what I mean by that is that there's a sphere of 3 nanometers on which 50% of the localization will fall, and a sphere of 5 nanometers where 90% will fall. So 1 nanometer sounds great, but actually has limitations if you're looking at protein complexes. And of course, representing is hard. You know, this may be not too flattering, but the data is very nice and it's clusterable. And if you cluster localizations and then use all the photons, all the repetitions to produce one single localization, it often goes in the sub-nanometer range, no problem. When you do storm, this is an origami, of course, but even in cells as well. And even I pulled out this data from the early times of MINFLUX, where we did accumulate all the storm localizations, all the storm photons, which, by the way, were 60,000, because we're not kicking them very hard. And well, then you can see here, this is 10 nanometer separation, and I don't know what the precision there is. Who cares, to be honest? A note on the beam shapes. You can do this with many beam shapes that have zeros. You can even do this with the Gaussian beams. With Gaussian beams, it gets more precise if you separate them more, because the tails of the Gaussians look like pieces of donuts. So if you use Gaussians, separate them. If you use donuts, put them together. What received a lot of attention was this case on which many groups were already working, because you can have a uniform distribution throughout your sample, but boost the precision that you would normally get on a camera by a factor of 2 or up to 2. So this was covered by an earlier paper, but then many groups took this over, and gave rise to improvements of this kind on the localization precision, which I think is nice. So, you know, mixing sequential structured illumination with single molecule localization is something that gave rise to very funny modifications of the resolutions and precisions. I think that's pretty cool. We have the MINFLUX. There's the SIM plus SMLM. I don't know how to call it. There are many acronyms for that. This is outdated, by the way. Maybe I don't have the right papers. Interactive MINFLUX also is another version of this, another flavor. And finally, MINSTED is a sequential approach as well, where instead of exciting with a donut beam, you excite with the effective PSF of a STED microscope, basically. So a narrower Gaussian. You can play with that. So bottom line, MINFLUX is a localization strategy. It is sequential. It is ratiometric. It's like a triangulation. And it uses finite points of a PSF in a smart way. MINFLUX is not a tracking strategy because the only job for MINFLUX is to tell you where the molecule is. It's not its job to tell you where you put your beam next. That's the job of a feedback controller. And there's a whole field that studies this problem. So if you have problems tracking, you probably have a not great controller, but it's not a MINFLUX problem per se. It is not power intensive. You use tens of microwatts. It's not easy to build. I'll show you now. And it's not trivial to process, because if you want to localize with 10 photons, you should not do maximum likelihood estimation. Happy to talk about that later. The necessary elements to play these geometric tricks are that you should have a spatial gradient of the mean and the variance of the thing that you're measuring. And happily, these photons that we measure are Poissonian, and comply with these requests. And finally, the non-necessary elements for all this is to make it fast, to make it single nanometer, or even single molecule. It can be a small object. It doesn't have to be fluorescence. Scattering photons are possible as well, though I don't see the point of doing something like that so far. Maybe somebody has an idea. It can be any pattern, and you don't even need a zero, because if you use Gaussian beams, this also works. So that's the bottom line, or the indoctrination part of MINFLUX. The six systems are a bit complex. The cartoons are silly. Systems are a bit more. They have more. We have lasers, beam shaping, wide field. Of course, you want to have as well. Multicolor single photon counting, stabilizations, and steering. Of course, fast electronics. Our current systems look like this. It looks terrible, but it's actually just an agglomeration of solutions to little problems. So happy to discuss this with the rest. Our daily activities have a lot of this. Very exciting. And these are the parts for a MINFLUX system, full MINFLUX system. So you can imagine we didn't bring this to Infomio. And the stabilization part, I'm happy to talk about it, but what you see here is peak-to-peak 10 nanometers of our measurements. And this is somebody just speaking next to the microscope. So you need to take care. We did some efforts on the 3D MINFLUX side because to steer beams in Z is not so easy. We originally used electro-optical lenses, which are terrible devices, to be honest. Never go for them, or if you go, just feel free to drop me an email. There are deformable mirrors, which are perhaps a bit costly and not the fastest, but we devised a solution with just focal cells and a lambda quarter, which is cheaper and faster. And what we did is to specifically work with this beam and use an interferometric approach. So most of the zeros of intensity that you see are a crossing of electric field from negative to positive. So if you overlap this beam with a constant beam, you can just displace the position of the zero, and voila, that's it. This that you see here represents what happens in the actual direction for this beam. And if you, of course, that's a nice representation, but if you compute this for realistic profiles of these beams, of the top hat beam that we use for actual directions, and a realistic Gaussian beam, of course, their coherent overlap also represents this, or is a good approximation for this, and you can use it to steer your beam in the actual direction. We built this with in the shape of a common path polarization interferometer that is based on a rotation module, a double pass on an SLM with a polarization rotation in between, and finally a polarization rotation fixed one and a polarization beam splitter. What this means is that we're polarizing the vertical polarization. Sorry, we're patterning the vertical polarization, we're patterning the horizontal polarization, then we rotate them both and make them interfere in a PBS. This is what we do. And you can regulate the relative energies of the two beams with a previous rotation. Happy to talk about that too, but it works well in a 600 nanometer range, which is more than enough for MINFLUX, and we put it to use in several scenarios. You may see that in the double pass on the SLM, there's some real estate available that we're making use of. I'll show you now in a moment. And what you see here is the transient of the deflection of the PSF measured with 400 nanoseconds time resolution. The extra real estate can be used for putting more beams there, so we can do multicolor, multi-shape MINFLUX, and we can also produce all sorts of other combinations that give you exotic beams, for which we have many plans. Actual tracking of MINFLUX look like this. This is a DNA origami that can oscillate actually, and we can measure it very, very precisely and very happily. We can even see transitions. We can see the transition is faster than 40 microseconds. That's not a surprise, but it's a nice bound. And for all these beams, what we're doing is multicolor 3D MINFLUX for multiple molecules, looking at conformational changes of some proteins. The protein in this case is cohesin, which is a large complex that is known to extrude DNA, and the mechanisms by which it does this job are not clear, and we want to dive in that. Today I will not be covering that. We're also working a lot of software packages for simulation, processing, and visualization, which is a big issue in MINFLUX. some outcomes of this is that if you use, instead of using a scheme like this, use a scheme where you switch the beams or change the beams, even canonical beams, you can have that for small L's, small patterns, you can have a factor of 2 extra or factor of 2 better localization precision, which can translate into 4 times longer tracking. So, if suddenly Luke or anybody came up with a dye that has 4-fold photon budget, this would be a big deal. But you can manipulate the beams to get that effect as well. So this matters, and there's further optimizations. Finally, we're working a bit on DNA paint that Luke introduced. I will speed this up. Until when do I have? What are the rules today? 10. Perfect. So we love DNA paint. Who doesn't? It gives these beautiful images. And we wanted to use it for MINFLUX. When I saw the work from Jörg's lab on the fluorogenic strands, I was very excited because fluorogenic strands means that you have a quencher in a dye and you can reduce background. And that's great news because background is the enemy number 1 of MINFLUX. So I wanted to use these strands for MINFLUX, but I also wanted to have multiple colors. And we said, okay, maybe we can create strands that are also orthogonal to those. But thinking about it, we also thought, well, what if we can create a handle that can also bind both of them? Because if I did that and I was doing colocalization with 2 imagers, I could, with 2 imagers, these 2 guys, A and B, I could localize 3 different handles by classifying. I could have the one that has only magenta, only cyan, or the one that has a mixture. And that would be great because if I use 3 images, I can have 7, and if I have 4, I can use 15, and the scaling up of number of targets with the number of imagers goes exponential. But not only that, another alternative to do this is to generate concatenations of strands that have different stoichiometries of binding to the different imagers. And in that case, it is possible then to have in between stoichiometries, and then with 2 imagers maybe have 5 targets. You can go even beyond the exponential scaling. So that sounded very appealing, but we had to do it, right? So we decided to pursue this by using the fluorogenic strands that Jörg described, and because I love this idea of the mismatches, and we thought, okay, can we redistribute these mismatches in a smart way, such that an imager and another imager that are pseudo-orthogonal, not fully orthogonal, have a good affinity for a pair and another good affinity for another pair, and in between one? Well, it turns out that you can do this, and you can do this analytically in a model, and then you can randomize it and do like large-scale searches by computing binding free energies. And it turns out that you can create sets of imagers and handles that follow this scheme. So you can have 3 imagers, 7 targets, and that actually works. You see here 6 of those targets. And the classification, the confusion matrices are very good. The origamis look wonky, but when we do MINFLUX on this, with this type of strands, it looks great. So these fluorogenic strands are longer than usual. There are 15 nucleotides, and they have affinity for the origamis in weird ways. So they look wonky, but then imaging looks good. Just saying. Now, what's the way to do the other case? Well, you just concatenate pieces of strands the pieces of handles that you know. So what you see here is the very popular imager, non-fluorogenic, and here you see one handle that has repeat sites to boost the K on. Here you have another one, and you can just concatenate pieces, and this just works. And you can have, for example, 9 targets with these with 3 imagers with the optimal stoichiometry, which is, by the way, and integers and then quarters, whatever. There's a whole optimization aspect. You can take the number of targets to 20 or more. And maybe Ralph will talk about that tomorrow because there's a coordinated development from their side as well. We made an army of reagents and we looked at fluorogenicities. Everything worked well. And the reason why we were looking at this is that I was very interested in RNA, as it was over there. So what you see here on the screen is RNA particles that were pulled from a lysate. And then this is an electron microscopy image. As you see, this is quite a heterogeneous object. And if you take one of these samples and make a tomogram by a tilt series, you can reconstruct the particles in 3D and even average them to find exquisite information about the protein complexes that are surrounding them. This complex here is the transport and export complex that is involved in bringing the particles out of splicing into the nuclear, close to the nuclear envelope to undergo export. And this is very nice, but does RNA look like a perfect sphere? I don't think so. But we don't know because we don't have a good technique to do that. So the question is there an underlying organization of RNA? If so, what factors are involved in regulating that? And which leads to the technical question that is, how can we observe RNAs in cells? RNA fish is, of course, the way, a good way to go, because you can label this dense object with sequence in a sequence specific way. You can couple this to DNA paint, because you can have addressability and high resolution and high detection rate, which is very nice for robustness. But if you want even more on this dense object, 3D MINFLUX might do well. So these two PhD students of my group, one of them shared with another group, embarked on this very ambitious task. And the first goal that we had with this was to try to correlate morphology of RNA particles with their gene architecture. So what happens if a gene is longer or it has more introns, because introns that are spliced out of mature transcripts receive a marking that is that is in the shape of a complex called the exon-junction complex. And these complexes are known to interact with RNA and with other proteins. So they may bring stuff together, or something might happen. So regular single molecule fish looks like this. And if you split your transcript into different pieces and image it in exchange rounds, you can have images that look like this, and you can have, the 3' to 5' distance as a proxy for a state. But what we want to do here is more on the side of this, where we of this, where we have the regular SM fish for a positive control, and then we do DNA paint with the fluorogenic imagers on the side. Fluorogenic imagers are quite important to get this to happen. What has been done before? Well, there's a paper from Ralph Jungmann, figure 4F or 5F in 2017. They look at this in a very large panel, but they didn't pursue further. In 2020, somebody developed a small RNA technique to do DNA paint by using exotic nucleotides to boost the K-on, to boost the residence time. And recently, there's been a MINFLUX paper where mitochondrial RNA was looked at, but no subtranscript information. So regular SM fish looks like that. The gene is this one. I'll say more things. If you do super resolution, looks like this. So dots turn into dots. Not very exciting, but if you zoom in, you can appreciate that there's some morphology starts to come up. So these are real shapes, or what we can measure of RNA, which seems quite rich. And if you do a few rounds, you can still start to see even more detail. So we chose a few transcripts to look at, 2 very long ones and 1 short one, and one of them has 100 introns. So if there's a difference, this should be visible. This is what these genes are in respect to the entire transcript space in terms of length and number of exons. And out of this class, we can get more. We want to get morphology. We want to get the end-to-end distance to compare to classical SM fish, what you will get out of that, and covariance and high order moments as well. The galleries of particles look like this, so not so easy to do by eye something. But if we look at the metrics, we can see that, for example, NDN1, this intron-rich and long transcript from nucleus to cytoplasm, has an elongation on the end-to-end distance, while the others do not have anything. Now, that's just end-to-end distance. If we look at the distribution, the covariances, we start to see that we see significant differences between nucleus and cytoplasm for the others. Aspect ratio shows some things as well. And interestingly, the kurtosis, which is a 4th order moment, like the covariance is a 2nd, this is a 4th, shows that from nucleus to cytoplasm, you go towards negative size, which is very prominent tails. So there are morphological features that are being rescued by this data. But I went on and on about MINFLUX. So what can MINFLUX look like in this type of sample? Well, initial data looks like this. So this is our intron-rich and large transcript in the nucleus, and this is what it looks like in the cytoplasm. We have several examples. So decompaction is very clear. Meanwhile, intron-less or transcript with few introns are much more decompacted, but still we do find statistical differences in their distribution. So of course we would like to get more data about this and get deeper. We're looking into metrics or representations of this information, kind of like the high C, so chromosome conformation capture metrics, because I think that may be useful. But all in all, gene architecture does correlate with the morphology. Of course, now we want to know what are the factors involved. Can we perturb them? Of course, this is something it's undergoing, and we want to see them alongside the RNA. Additionally, we would like to do morphology-inspired probes to perhaps label the proximal sites to the exon junction sites and see what happens there. On the technical side, we want to push for tracing more finely. I think it is possible to trace RNA way more finely and to see more RNAs, hundreds of them, to paint a more coherent picture. And of course, we need higher throughput, which is also on the way. But stepping out, I want you to reflect on where are we coming from. Standard or regular SM fish looks like this from the 80s, from Robert Singer and others. But of course, nowadays with the best probes, the best cameras, the best protocols that we have, the best probe design, the probe design is key here for all this that is shown. But we can already, without MINFLUX, we can have a new set of eyes on this object, and even with MINFLUX, take it to the level of looking what these flexible objects are actually doing in several scenarios. Because there are many corners of RNA biology that have remained untouched. Of course, there's RNA-seq and there's MERFISH, where you strive to know the identity of RNA transcripts, but you don't get information about their shape and interaction. Some sequencing novel techniques are doing that, but then you lose the information about where in the cell this is happening. So I think there's a bright future for this new way of looking at RNA in transcription, in translation, long non-coding RNAs, RNA viral packing, and so on. So with that, I want to say some words about where I am. I'm at the Vienna Biocenter, which is a very biological campus with thousands of people, 160 groups. If you're interested in interdisciplinary biology, this is a great place to be. Take a look at the websites. And then I want to acknowledge my group, especially Alessandro and Paul and my collaborator Clemens, which we have been doing together this RNA story, and the rest of the group for everything else that you saw. With that, I hear your questions.
Thank you so much for the wonderful talk and the wonderful movies. Just to start off with kind of a general question. So we're talking about at the end, you mentioned doing RNA fish, DNA paint, and MINFLUX all in one go. Can you give us an idea of like the amount of time it takes to prepare the samples, do the acquisition, do the processing?
It's tragic. So DNA paint is not the fastest in the neighborhood. MINFLUX not. So you can imagine the combination of them, it goes slower because you're looking at single confocal spot at a time. So DNA paint rounds here. So if we do wide field DNA paint, so let's go in order. RNA fish protocols are overnight. So we do that, and if we do proteins on top, sometimes it's day and a half. So the protocol is a bit long, but it's okay. To be honest, it takes longer to design the probes than to do the protocol. For the acquisition, wide field data is just 20 minutes per round. So if you do 3 rounds in an hour, you got a cell. I think it's okay. It's at the RESI level, or this type of things. Then if you do MINFLUX on it, you do need to stay. So you get, say, 3 particles per hour, something like this. Very slow. That's why for DNA paint, we have few things. And for wide field DNA paint, we have hundreds of particles. Not 1,000, not 10,000. So I think what will happen here, of course, there is a need, not for this, but for other reasons as well, to have these high-end structured illumination-based techniques boosted in throughput. And several groups are attacking that, of course, me too. And it will be important to do relevant things with them.
Hi. So in this condensing, was it that would you show?
Cohesing was it?
Cohesing. So
it was, yeah.
So what is the? I'm probably you mentioned, but I missed it. So is that on cells or is it on purified protein? And what's the kind of resolution you can get in time?
So this is cohesing. We're putting cohesing with or without NIPBL, with or without ATP, and with or without DNA. We have cohesing with 5 orthogonal sites that we can label modularly. What I showed is just 2 of them. Let me pull up this. It should be somewhere. Mm-hmm. Oh no. Here. Maybe this. And what you saw there was just 2 sites labeled down here in the heads with a halo and snap tag and 2 dyes. Now,
it's immobilized.
This immobilized is in vitro and is purified. We're not
resolution.
Sorry.
The time resolution.
Time resolution depends on what spatial resolution we want to have, because you have a trade-off there, and you can choose in post-processing what you want to do. We are normally in the 3, like sub 10 milliseconds easily. So we don't need more because things are not so fast. We care more about where in 3D both molecules are. And of course, we have in the pipeline to put more molecules in there. Our goal here is to first identify the conformational changes and their kinetics with and without certain partners, and one day do this on top of an extrusion assay. One day.
Thanks for the great talk. So obviously for MINFLUX, having a lack of photons is incredibly important.
A lack of?
Photons is incredibly important for localizing your signal. Maybe this is a silly question, but how do you handle differences in fluorophore polarization and orientation and or resonance energy transfer when you've got 2 spectrally overlapping fluorophores that can also have an absence of photons, not from the center of your MINFLUX donor?
So as you might well know, the polarization of the beam at different locations is very different. That is especially true for the top hat beam. The top hat beam in the lobes, in the axial lobes, is X polarized, and on the lateral lobes is Z polarized. So if you have a fluorophore or a dipole that is static and you do MINFLUX with this beam, you're going to be probing it in a bit of a weird way. So actually, if you mix top hats and vortex beams, vortex beams only probe the XY and don't probe in Z, mostly. So that's a way to escape the, let's say, differential axial lateral probing of the top hat only strategy. So, but for that to manifest, you have to have a dipole that is not moving, which I never see it, to be honest. So I don't care so much, but we are aware.